every year I'm always surprised at how well the students do in finding both the breadth and the depth of their projects, the diversity of projects, and their quality. We're going to try and keep more or less on a schedule. Students will be presenting for about 12 minutes, and we'll leave a few minutes for questions. I think the best way to ask questions is through chat. You can just add a chat note saying you have a question. You don't have to tell me, and then I'll call on you and you can ask your question. So without further ado, I'd like to introduce the first speaker, Nolan Slattery, who's going to talk on the New York State Marine Aquaculture Prospectus of 2020.
Okay, so Marine Aquaculture Prospectus of the New York State 2020. So my capstone project was essentially to produce a prospectus on the state of saltwater or marine aquaculture in New York State. The industry is still quite new. Developments in technology and regulation all over the world are having significant impacts on the global seafood market, with obvious implications for conservation and marine resource management. Regulating authorities of aquaculture in New York include the various townships with aquaculture, with shellfish populations such as Islip, Oyster Bay, and Huntington. For the sake of this project, I focused on the Suffolk County's aquaculture shellfish lease program, under which I was assigned a commercial shellfish lease in 2018, so I have some personal experience with. Marine aquaculture can be classified as either coastal or offshore. Nearshore aquaculture, also known as coastal, is best suited for shellfish production in its current state. Shellfish are naturally occupy these shallow waters close to shore. Offshore is also being pursued for the promise of finfish aquaculture, whereby the huge currents and large water depths allow for a lot more flushing of potential waste from the finfish. While offshore aquaculture can include shellfish that would naturally reside in that ecosystem, such as sea scallops in New York. The momentum behind the sector is definitely predicated on the commercially valuable finfish. Offshore aquaculture in federal waters is gaining momentum. It could make its way to the United States and eventually New York. The offshore aquaculture is managed by the U.S. government of various federal agencies. That area begins 3 miles offshore and extends to 200 miles offshore, 197 miles. It's called the exclusive economic zone. You can see here on the green line. Everywhere else in this peach area here is managed by New York State. In 2009, it became completely official in the current state, the aquaculture program, where Suffolk County is in charge of all this area east of Riverhead, so Peconic and Gardner's Bay estuaries. So a history of aquaculture in New York. So according to the Food and Agricultural Organization of the UN, aquaculture is the farming of aquatic organisms in both coastal and inland areas, involving interventions in the rearing process to enhance production. So according to the FAO, aquaculture is the farming, in 1715, a bill was introduced in New York that prohibited the harvesting of oysters in summer months to allow for the oysters to repopulate themselves naturally. This early attempt at aquaculture, as defined by the FAO, as an intervention of the rearing process, was actually petitioned in 1807, as seen here in this screenshot of these are two different documents, found an index of economic materials from New York State. 1807, the penalty for taking of certain beds of oysters. And the petition was gained enough momentum, they were able to overturn that law so that they could harvest oysters in New York all year round. And it was basically based on popular demand because the oysters were so sought after at the time. In addition to 1807, you can also see statutes dated in 1831, an act to restrain the taking of in Hudson River. In 1840, an act to prevent the destruction of, all related to oysters. So you can see there's some history there regarding oyster regulation. And that reversal in 1807 coincided with reports from Connecticut, as well as Great South Bay, of increased poaching at the time based on that increased demand. So basically, the poaching got so bad, the restriction of harvesting in New York Harbor not allowed, but encouraged the oyster shell fishermen at the time to go out to other beds in Connecticut and Great South Bay and start poaching on the beds there. So they basically opened up the beds of New York Harbor. As New York Harbor continued to industrialize in the Hudson River, the Hudson River estuary oyster beds were compromised, primarily due to the sewage inputs. The sewage system, I think, in New York was installed in 1849, and over-harvesting also had an effect in some areas. That led to the proliferation of oyster aquaculture in regions close enough to New York to serve the market with less compromised water quality. So in 1884, New York State, recognizing this, officially ceded the underwater lands of Peconic and Gardiner's Bay to Suffolk County for purposes of developing legislation to promote commercial aquaculture in the water, in these waters. And the industry thrived, albeit briefly, as the supply of oyster seeds used by the Long Island farmers began to diminish in the 1990s. The original state law had a number of enforcement issues regarding lease assignments and ownership in 1884. So basically, the way that they were managing the aquaculture at the time, the program combined with not just the aquaculture, but the shellfishing. So just the shellfish harvesting at the time, and the worsening environmental conditions as well, all combined basically to hurt, kind of wipe out the oyster industry by let's say 1920s, 1930s. But over that time, the early 1900s, there were a number of achievements made that would enable the county to eventually, the county law to eventually be codified by the DEC in 2014 by the Environmental Conservation Law of New York State. That helped. The amendments were made to the law in 1884 in 1969, and those included the ability of shellfish leaseholders to farm additional species other than just oysters, as well as turn the language from assigning a privately held grant, to a lease, which made it easier for the county and state to basically reclaim the land if it wasn't being used properly. Also, there was in 1923, there was an algae cultivation method that was developed in Suffolk County that's was adapted to the Milford method, which is still used today. And all of these trials and tribulations, so to speak, led to the 2004 codification of the law by the DEC, which put stipulations in place, will help to accomplish the stipulations that were in place on the state's original secession to the county in 1884, such as the completion of a variety of environmental impact statements that they were not able to accomplish until 2004. Once those were accomplished, the county was officially able to adopt their program in 2009. So that took the program to as we know it. The lease program ran from 2009 to 2018, at which point, in accordance with the county's original plan, a 10-year review began that's ongoing right now. So the shellfish cultivation zone is shown here in green, which is the area within the county's jurisdiction that was certified to authorize leases. You can see the navigation channels excluded, these thin kind of lines woven throughout, as well as areas that are not typically safe for harvesting of shellfish farther west to the estuaries by the mouth of the Peconic River. Since the 10 years farms have begun to expand and alter the equipment and processes used throughout the cultivation of shellfish. Like I said, it's like still a very new industry. So people are kind of trying different things out and figuring out what works in this area. Competing stakeholders, however, have begun to push back on the trend, such as the floating shellfish gear. So as part of the 10-year review that's ongoing at this moment, public outreach sessions are held, as well as the formation of an advisory committee with representatives from all the various stakeholder groups throughout the bay, sailing and recreational fishing associations, commercial fishermen and shell fishermen, relevant town legislators and county representatives. The town review could alter the terms for renewal of existing leases, as well as make changes to the program for future lease cycles, such as altering the shellfish cultivation zone, that's the green zone here, in light of the feedback received during the 10-year review. So for instance, here's a screenshot of one of the 6 alternative maps. I know it's just a small portion here, the southeastern corner, but it's to show here the changes in acreage. And also this specific map was, I believe, Amendment 3. It's basically depicting in yellow all the areas that were existing in the shellfish cultivation zone that would be prohibited from using floating gear. So aquaculture could still occur in these areas, but it would have to be not on the surface. It would have to be submerged at the bottom. So it would, diminish the area of where you'd be allowed to use the gear, as well as the total acreage due to some of the DEC changes as well. So the reason for this change to the shellfish gear is that opponents of this gear type say that when the industry was introduced in 2009, bottom gear was represented as the dominant form of equipment used in the industry. And since that time, more and more floating gear can be seen on the water, which occupies a significantly higher footprint than bottom gear, and diminishes the usability by other stakeholders. So this basically has led to a debate over the term hazard to navigation that's seen in the Environmental Conservation Law of 2004. And that debate's been ongoing. And so it is a big deal to the stakeholders involved, how the county handles the 10-year review, especially the use of floating gear, as that's like the issue of most concern to stakeholders in the bay. So internationally, aquaculture has grown quite a bit, as we all know. Seafood import statistics are very difficult to accurately measure, however. So there are estimates that the U.S. imports between 60 and 90% of its seafood, about half of which is farmed. The discrepancies between about 62 and 90% can be attributed to conversion errors, as well as increasingly global supply chains. Some believe that the 90% figure basically does not account for seafood that is captured and consumed domestically in the United States, but maybe processed to some extent overseas. So maybe the fish will be captured whole here in the United States, frozen, sent overseas to be, scaled, filleted, processed, and then sent back here. So, where exactly does that fall in terms of, imports, exports? That would obviously have an influence on the 60 to 90% number. And then in terms of the conversion ratios, it's difficult to convert, in terms of consumption or how much a country's importing, exporting. Some of that might be canned, some of that might be processed to, all different varying degrees. If it's a whole fish, if it's mixed with other ingredients, things like that. So it all complicates the numbers. But
Nolan,
with that, yes.
one more minute.
Okay, shoot, sorry. All right, so that's basically this growing trend in aquaculture is leading to the Blue Revolution. The Blue Revolution, quote unquote, basically that as the global population continues to rise, we're going to need to increase aquaculture production to meet that demand. The things that might help facilitate this would be that offshore adjacent industries like oil and gas and wind farms, they have the infrastructure established offshore, as well as a lot of the technical expertise to work in these very harsh and difficult environments that could be used in offshore aquaculture. However, could be very controversial. Environmental groups and commercial fishermen have given a lot of pushback to aquaculture historically. beginning in 2005, there were various movements in Congress to enhance aquaculture. They were all denied until 2020 when an executive order was issued. So obviously there's no, we don't know exactly how that will play out. But NOAA, the idea of the executive order is to appoint NOAA as the lead agency, for which there's no lead agency in aquaculture of offshore waters at this time. The main takeaway is that we need clarity in legislation because there's a lot of inefficiencies occurring in the industry right now. So aquaculture in New York will definitely be influenced by the results of the executive order, as well as the 10-year review process. Hopefully, as the county resolves its process and the executive order plays out throughout the various levels of government, clarity will be established to help protect our marine resources for the future in the way that's going to benefit, you know, our marine ecosystem in a way that can enhance and solidify the value that we're able to produce that we rely on from our marine ecosystems. All right, thank you very much.
Thank you. There's your applause.
Thank you. Should I stop sharing for questions or no?
We have time for one quick question.
Okay, sorry about that.
Any questions? I have one. You didn't really discuss what species are dominating New York aquaculture.
Yeah, so species, it's definitely the oyster. Secondly, it would be the quahog clam, and the bay scallop is gaining a lot of momentum, particularly since 2019 had such a horrible bay scallop season. However, Cornell Cooperative's bay scallop sanctuary in Orient Harbor, they had a much higher survival rate inside of their lantern cages. I would speculate that has to do with they were protected from the water as well as to harsh water and predators, so there was less stress on them, and maybe that was able to had more resources to survive the low oxidation and increased water temperature.
Okay, thank you, Nolan.
All right, thank you.
The next speaker is Amanda Gorman. She'll be talking on the temporal water quality analysis of Virginia Beach watershed. Amanda.
Hi, everyone. All right, so I'm going to start, I guess. Hi, my name is Amanda Gorman, and I'm going to be presenting on my paper, The Temporal Water Quality Analysis of Virginia Beach Watershed. So here I have an outline of what the presentation will look like. I have a brief introduction of myself and what my internship looked like, a list of parameters tested. Then I go into a site overview, and after that I get into some data that interested me the most and makes up the bulk of my paper. It's not everything that I covered in my paper, but that would take way too long to go over. And then I'm following that up with a discussion on why we might be seeing this data, some future study questions, and then finally I connect that all back to marine conservation. So I've kind of got a lot to cover in 12 minutes, so let's get started. So just a little bit about me. My name is Amanda Gorman. I became interested in marine conservation issues while writing my undergraduate thesis on haplotype diversity of a sea cucumber species that was declining in population around Belize due to overfishing. And this summer, I did my internship with the Virginia Aquarium and Marine Science Center. I was one of 3 interns in the water quality lab, and I worked under the supervision of Allie Hobgood. Dr. Bruce Brownwell is my academic advisor. The internship ran from July 1st until August 13th when I gave a presentation to the aquarium. We did field sampling and lab work from July 7th to August 6th, Monday through Thursday. This research was conducted in partnership with the Virginia Department of Environmental Quality. They receive our data and will determine if future testing or intervention is necessary. So these are the parameters we measured at each site Monday through Thursday in the morning. Anything underlined is something that I will touch upon this morning. Due to limited time with COVID and only having 2 weeks to analyze the data after collections. I did not do any statistical analysis of the data. My advisor and I spent our time looking for observable patterns and trying to figure out why we saw what we saw. But in the future, that would be an important next step. So I wanted to kind of start off with some pictures of the sites. We tested 11 sites total, sites A through K. Visually, in these next few slides, you can see differences in things like width, depth, water color, and turbidity. All sites are relatively shallow. One site had an average depth of about 3.3 meters, that's site C, and that's as deep as they go. The other sites' averages are between 7 and 65 centimeters, so they're super shallow. And they're also freshwater. They are kind of more inland sites of these rivers. So really quickly, I'm just gonna name these sites left to right and give a little bit of time to just look at the pictures. So from left to right, it's A, B, C, D. And then we have E, F, G, and H. And then we have I, J, and K, which you can barely see at all because, well, we couldn't either. So next, I used GIS to georeference a land use map image onto a coastline map with coordinates to observe where these sites fall on that land use map. All sites are either in residential or commercial areas. It's a little hard to see because the points had to be big enough to see. But 3 sites exist right on the border of woody wetland areas. And as I said, it's a bit difficult to see, but they do all fall into a mix of residential and commercial areas. Then on the right, I also put together a quick map on Google Maps. So this shows to the right the Virginia Aquarium and Marine Science Center. We started setting up at the aquarium at 8 a.m. and then we sampled in a circle, A through K in that order. So, you know, we went down and around sort of. And that affected when we sampled them, obviously, because we sampled A first every single time. I used color to show the connectivity of these water bodies. Site A is an unnamed tributary to Red Wing Lake. B is an unnamed tributary to West Neck Creek. Sites C, D, E, and F are all locations on North Landing River. Site G is an unnamed tributary to the North Landing River. And sites I, H, and K are all unnamed tributaries to London Bridge Creek. And site J is an unnamed tributary to Talia Creek. Following all these streams, it appears that sites I, H, J, and K eventually make it into the upper Lynnhaven Bays. Sites B, C, D, E, F, and G flow downwards into Currituck Sound in North Carolina. And site A flows into Red Wing Lake, which looks like it might also have a connection to the ocean. So at least 10 sites out of 11 have the potential to impact marine environments, and that's the basic point I'm trying to get at there. So I wanted to touch on this super briefly. Sites had average dissolved oxygen percentages that were below saturation, probably due to in situ respiration in the sediments. This led us to believe that the sediments could possibly be a source of ammonium and phosphate. We only tested in the morning, so we only got a really vague snapshot of morning oxygen levels, but most sites were below 50% dissolved oxygen saturation. So Enterococcus species are a common fecal indicator bacteria used to predict pathogen levels in water. We used Enteralert for our measurements. This system gives us an Enterococcus measurement in most probable number of colony forming units per 100 milliliters of water. It's important to note that Enteralert detects many species of Enterococcus, not just species related to humans. Enterococcus is a common bacteria in the intestinal tracts of warm-blooded animals. There have also been studies showing the presence of naturalized colonies that persist in sediments or on submerged aquatic vegetation. Enteralert picks up species from humans, many mammals, birds, and these aforementioned naturalized colonies. It does not give us a source, but it's still a good test to perform for frequent monitoring. So before analyzing the data, I hypothesized that lower depths would have higher Enterococcus levels. Our findings were, well, sometimes. Sites F and G and K, the shallowest sites, do have really high Enterococcus levels, but other sites don't necessarily follow this pattern. We can't definitively state that depth is a major factor, only that it could possibly be a factor of many. So does depth impact Enterococcus levels? Probably. Given equal input, it would make sense that more water would dilute Enterococcus. Both of these charts that I have here are depth and Enterococcus levels plotted by site, but the one on the right does not include site C, just so we could see the other points a little bit better, because site C was so different in terms of depth. So then on to the next one. These are all site-specific charts that show precipitation levels and Enterococcus levels. I had predicted that Enterococcus levels would increase after heavy rainfall, and as you can see, not quite. I included sites A and G to show general variability, but if I can draw your attention to the right side of the screen, on the bottom, we have site K, which had massive Enterococcus increase after our largest precipitation event. But if we look up to site F, the first big spike in Enterococcus happened a week after a rain event, presumably from other factors. Also, I want to note how high these levels are. All 4.5-week averages of Enterococcus levels are above 100 most probable numbers of colony forming units per 100 milliliters of water, and some are in the thousands. The EPA has set a recreational limit of 35 colony forming units per 100 milliliters of water, and our data predicts much higher values, and I will get into that more later in the discussion. So for our nutrient panel, we tested ammonium nitrite, nitrate, and orthophosphate. It's useful mainly in the prevention of algal blooms. High levels of nutrient pollution can cause harmful algal blooms. Ammonium, nitrate, and orthophosphate are the 3 tests that I will be looking at closer. Nitrite values were super low and didn't really show much of a pattern that I can see at this point in time. So here I explored 2 sites, F and G, that show similar nutrient patterns in relation to precipitation. While nitrate levels differ, probably due to source differences, their highest levels of ammonia and orthophosphate are after the biggest rain event. So that was exciting to see. But next, I show a site that doesn't show this. So comparing site J to site F, J had a slight nitrate spike after the rainfall, but both ammonium and orthophosphate levels are relatively consistent and pretty low. So this further illustrates the variability among sites. So from our results, we can see that many variables affect Enterococcus and nutrient levels. While precipitation and depth are possibly factors, it's hard to gauge their impact due to the highly variable data. Obviously, as most studies go, we had some limitations. Time was a big one. More data would give us more to work with, more rain especially, because it didn't rain all that much while we were testing. And that would make it easier to determine any patterns. Another limitation is that we don't know the sources of Enterococcus or the nutrients. And so I have some future questions that would provide more insight. And first, a good question is, what is in the sediments? The ones that I stood in were muddy and smelly, and we assume that they are nutrient dense. But it would also be useful to see if Enterococcus is persisting in sediments to see if the resuspension of sediment could be impacting our levels. A qPCR analysis would be useful to determine species of Enterococcus so that a source can be identified. And it would also be nice to get a better grasp on flow. Most were super low flow, almost stagnant, but post-hurricane, the flow increased at most sites. So it would be cool to see how much these streams impact larger water bodies over a longer period of time, because we didn't measure that. So I go back to the importance in marine conservation. So this type of testing can possibly lead us to where the nutrient or bacteria sources are located. Similar data can be used for harmful algal bloom prevention, and in our case, our data can be a starting point to further testing. The Enterococcus levels we are seeing are well above the EPA limits for recreational water bodies of 35 colony-forming units per 100 milliliters of water. Should this Enterococcus be carried to bigger water bodies, the associated pathogens could impact shellfish industries or recreation. And in this case, further testing is really necessary over a longer period of time to determine if what we're seeing is accurate, especially with qPCR will be necessary to determine where the sources are. Yeah. So does anyone have any questions?
Questions? We have a bit of time.
Amanda, this is Bob Serrato.
Hi.
Does Enterococcus survive in seawater?
It does. I think it survives longer than E. coli, I'm pretty sure. That's why it's becoming more of a standard for seawater testing. As far as freshwater goes, I think E. coli is still technically the standard for fecal indicator bacteria, but it's becoming the standard for fecal indicating bacteria of marine environments.
Thank you.
No problem.
Hi, Amanda. It's Ellen.
Hi.
I'm just curious, how did you line up this internship with the Virginia Aquarium and Marine Science Center? Was that through your own personal connections or some other way?
Honestly, I looked it up online. My parents live in the area, so I was just looking up stuff around Virginia, and I found this internship and applied. And I really like this internship because we were doing our own sort of project. It was like this, we went out by ourselves in a van and did all the sampling and came back and analyzed everything. So it was really, really cool experience.
Yeah, that's great. Thanks.
Any other questions? If not, okay, thank you, Amanda.
Great.
Please stop sharing your screen.
All right.
The next speaker is Dickens. The title of her presentation is Searching for Rachel Carson. I really like that title, by the way.
Thank you, Glenn. And happy retirement, I think.
Almost.
Let's see here. Can you see my screen?
Yes.
Great. I'd also like to quickly thank Ellen Pickett for your encouragement, and I'm also very grateful to Carl Safina for your support and mentorship this summer. So my name is Hope Dickens. I am interested in communicating science, and so I spent the summer researching and writing long-form essays covering a range of topics related to environmental conservation. Those writings included personal narratives, opinion pieces, and reported articles. One of my articles was published just last week by the environmental news organization, The Revelator, which you can see a screenshot of here. The Revelator is an initiative of the Center for Biological Diversity, so I encourage you to go check out that article. Today, I'll be reading a shortened version of an essay I wrote that reflects on the life of Rachel Carson, who is the main reason I pursued marine conservation in the first place. It's called Searching for Rachel Carson. I discovered Rachel Carson later in life than I'd like to admit, but when I did find her, she hit me with a bang. Who was this writer that could weave a tale of water, animals, and shoreline as if they were characters in a dramatic novel? Who could transform the history of the planet from its typically dry, humorless account into a tale of vivid awe? Who was this woman? I would discover that while her writing held me spellbound, it is her life story that contained abundant inspirations. Rachel Carson changed the world. Dutifully, humbly, She devoted the last years of her frail life to a topic she wasn't particularly passionate about, pesticides, but whose presence threatened that which she was, the natural world. Without her, we may well have lost songbirds and bald eagles, insects, and each other. Rachel Carson wrote a book, Silent Spring, that all Americans could read, not just the scientists, not just the intellectuals. The farmer, the housewife, the school teacher, they could read it, and they did, in droves. She articulated for them what they were already noticing in their backyards, and beyond that, she articulated the instinctual knowledge of interconnectivity with other living things. She taught everyday Americans what an ecosystem was. She introduced to the masses the concept of ecology. As I learned about Carson's life, it was easy to be impressed by her accomplishments. Any person that spends years researching a complicated subject like pesticides and writes it into a book comprehensive enough for the layperson, and in doing so takes on the chemical industry and changes the minds of a president and everyday Americans, and in doing that is credited with the banning of harmful insecticides and is therefore viewed as the figure that launched the modern environmental movement, is a person to be lauded. What stirred a deep admiration for me, though, were the parts of Carson's life that were out of the public eye, the parts that she took pains to conceal, and the parts that were distinguishable only if one were to read between the lines. Carson's bad deck included not just being born female at a time when so few opportunities were available to women, but was also comprised of responsibilities and illnesses associated specifically with her gender. As the only surviving daughter in her family, and at a time when male peers of lesser intelligence were being ushered into prominent doctoral programs, the duty to provide money and caregiving for her sickly mother and orphaned adolescent nieces fell directly on her shoulders, eclipsing her career aspirations. Later, when she was able to pursue a career as a writer, she was not only tasked with providing daily care to her now dying mother, but was again charged with the monumental role of caregiver to her orphaned grandnephew, legally adopting the boy when he was 5 and she was 50. To complicate her life further, Rachel Carson was frequently sick. Having already had one cancerous lump removed from her breast at age 43, she was again beset by breast tumors at age 53. Lying to female patients about the truth of their medical conditions was a common practice at the time. So when Carson, post-op, asked if her removed tumors were malignant, she was told no and was recommended no further treatment. The truth was they had already metastasized. The fact that she was lied to about her own body undoubtedly delayed the radiation treatments she would later receive when it became obvious that she was seriously unwell, and thereby hastened her death. The critical and public reception of her writing was also tangled up with her womanhood. When The Sea Around Us was published, it was an instant triumph, but its popularity and acclaim did little to stem the sexism from male readers and critics. The New York Herald Tribune assured its readers that Carson was an attractive woman, and the New York Times reviewer lamented the lack of a photograph of Carson on the book jacket, as though her appearance would qualify her credibility. With the publication of Silent Spring, the gauntlet of sexism was much more brutish than benign. Despite the 55 pages of principal sources that Carson lists in the back of the book to offset any suggestion that her research was incomplete or biased, scientists and lobbyists aligned with the powerful chemical industry attempted to discredit her by running the gamut of insults, from her appearance to her age, to her credentials, to her status as an unmarried woman. They poured money into marketing campaigns designed to invalidate her intelligence and comprehension of insecticides, which backfired splendidly by drawing more attention to her book and thus increased its sales. It took Carson 5 years to write Silent Spring. She did so while tending to and suffering the loss of her mother, while adopting and becoming a full-time mother to her grandnephew, and while enduring constant medical setbacks. What resonated with me about Carson was not that a woman could earn the accolades and support of men at a time when so few did. What resonated with me was that she did so while overcoming incredible odds. What would the world look like without Rachel Carson? And who would I be without her? I want to be a woman who communicates science in an unscientific way. Rachel Carson showed me it can be done. Since learning about her, I've longed to know her better. So I set out on a pilgrimage. The first stop was the Rachel Carson Salt Pond Preserve in Bristol, Maine. In the edge of the sea, Carson says, quote, "The pools have many moods." And as I traipsed from the sun-drenched high pools to the seaweed-filled lower pools, I saw what she meant. The high pools were harsh, hotter, saltier, more barren, less inviting. They were the deserts of the rocky shore if one could imagine earthly habitats on a miniature scale. And like the desert, their purest elements, light and color, were their most seductive. The lower pools were jungles, absolutely alive, their bizarrely shaped seaweeds swishing with each rush of the current, shielding countless microbeings within and beneath them. Because of Carson, I knew to step gently and look closely. I knew that even the smallest depression in the rock held living beings, and I couldn't help but marvel at the life forms that managed to find their way to the narrowest strip of water within the primordial grooves of the stone. Next was her cherished island of Southport, Maine, where she spent her summers. At the southern tip of the island is the Norwegian Seaside Inn, a place frequented by her and her close friend, Dorothy Freeman. I walked the short path from the inn to the sea, the same path Carson took to the 2 Adirondack chairs where she and Freeman sat summer evening after summer evening. Just below that, as the forest gives way to shoreline, a bronze and copper plaque is affixed to the smooth, flat face of a boulder, memorializing the place that Freeman spread Carson's cremated remains in 1964. As I read its words, quote, "Here at last returned to the sea," I felt an emotion akin to grief-filled gratitude take over as I looked out at the ocean and thanked the spirit of the woman so far away in time and yet so present in every wave lapping against the shore. A year has passed since then, and largely thanks to Carson's influence, I am on the cusp of receiving a master's degree in marine conservation. To bring my understanding of her to its fullest and to complete this pilgrimage, I visited her home in Silver Spring, Maryland. The modest house is on a corner lot, tucked behind a large pollinator garden and stands of trees. Its owners, a kind, elderly married couple and retired scientists committed to upholding Carson's legacy, invited me in. Her picture windows are just as she described them in her letters to Freeman, large and looking out on her little wild plot of land. Her study is large, wood-paneled, and warm-feeling. Her writing desk is tucked into the corner where the windows let in leaf-filtered sunlight. I stood over her typewriter, the very place she wrote Silent Spring, and felt like I was touching the transcendent nerve of greatness, a sort of environmentalist mecca. I've glimpsed Carson's life, the tidal pools, her study, her bedroom, her resting place, and yet she is still unknowable to me. I've stared at pictures of her, class photos, family photos, press photos, and in searching her face, there is still a mystery, a deep well of wisdom and of sorrow behind her eyes. Her letters to Freeman are as close as one can get to knowing her heart, and even then, the most heartfelt of them were destroyed by the two women in a pact to keep her innermost thoughts away from prying eyes like mine. The not knowing is a gift. By keeping her life private, but offering us her research, her prose, her words imbued with the tenderness of a woman charged with caring for life young and old, human and non-human, she has created pockets of potential for women like me. Women like me who are eager to fill the spaces between science and emotion, between knowledge and awe, like the ocean rushing into the ever-changing tidal pools, constructing endlessly dynamic and mystifying futures still to be discovered. Thank you.
Thank you, Hope. That was very nice. We have time for some questions.
I'm speechless. That was fantastic. Thank you.
Beautiful.
Thank you.
That was superb.
Thank you. Thank you.
Hope, you mentioned you wrote several long form essays. Can you just mention what the topics of some of the other ones were?
Sure, yes. So I wrote an essay in the wake of the Black Lives Matter movement and the killing of George Floyd about, let me just gather my thoughts. It was a personal reflection on the racial disparities in the environmental and conservation world, all the way from birding to publishing. I also wrote an essay that used the occurrence of the pandemic to investigate the sort of psychology themes of crisis-inspired reactions. So I looked at phenomena like the increased philanthropic giving, the decline in roadkill, and then an uptick in citizen science participation. And from that essay became a shortened essay that was published about the impact of on wildlife rehabilitators during the pandemic. So that's what I wrote.
Thank you.
You're welcome.
Hope, do you think, are you thinking about places you might be able to publish this essay?
Yes, and in fact, the 58th anniversary of Silent Spring is coming up next month. So I'm hoping to shop around the longer form of this essay to as many media outlets as I can. So I'll keep you posted if that gets published.
Anybody else? If not, thank you, Hope. And we'll go on to the next speaker.
Thank you.
The next speaker is Kayla Clauson, and she'll be talking about horseshoe crab assessment of Prudence Island, Rhode Island. Rhode Island, excuse me.
So I am Kayla, and I'm going to be discussing my assessment of Prudence Island horseshoe crab populations and the associated parasitic interactions. So first, I just want to explain where even is Prudence Island in Rhode Island. It is the third largest island within Rhode Island's Narragansett Bay. It's situated within the geographical center of the bay. It stretches about 7 miles long as the crow flies and about 2 miles at its widest part. It's not the biggest island, but it is home to just under 100 year-round residents, and 85% of the island is protected from development through state and other conservation agencies, as you can see in this map here. Other nearby islands that fall under the state conservation include Dire, Patience, Hope, and even Despair, which can only be seen at low tide off of Hope Island. So luckily for me, I was able to do my internship through the largest conservation group on the island, through Narragansett Bay National Estuarine Research Reserve, or like we like to call it, NEBNR for short. NEBNR is funded through Rhode Island's Department of Environmental Management, along with NOAA and Rhode Island's Audubon Society. My primary roles were doing education and research. For education, there was a lot of it was with homeschool curriculum development, with the homeschool groups that would visit the island twice a month. And here is me with one of my groups planting salt marsh cordgrass or Spartina plugs into one of our marshes. Another primary education chunk of what I did was family programming, such as having island families and visitors explore our shores and do activities such as the one photo here of dissections. The other main part was research, and they do a lot of ecological monitoring on the island. A lot of the research has been going on for well over 10 years. So I was an assistant for this year's data collection, such as biweekly osprey surveys. There's 10 breeding pairs known on the island. Invasive Asian short crab surveys, and breeding songbird surveys, which you have to be really early in June to do. And I'm lucky enough to be a part of the pilot program for wildlife camera traps, which we just started about a month ago. We have cameras set up within 3 of the habitats on the island, and just to get a general idea of the animals that are using the island, such as this little coyote here. For my individual research project, I wanted to look into the horseshoe crab population on Prudence. It has not been yet quantified there. The island is only accessible by ferry, so it definitely constrained the researchers' hours and didn't work out with them to monitor during the nights. The last ferry off island is at 6 p.m. However, I did live there, so it was easy for me to do, and it just worked out because they are one of my very long list of favorite ocean creatures. So the horseshoe crab population assessments are needed in order for management and protection policies to be decided. Oh, why did that change? The Atlantic horseshoe crabs are near threatened, mostly due to overexploitation for bait fisheries such as eel and whelk fishing. Also, they are really important in the biomedical community for their use of their blood and vaccination development. And I also wanted to quantify the target species parasites. In wildlife biology, parasites are often overlooked, sometimes because they're just difficult to study, because they're often pretty small and they have complex life cycles. However, this parasite, Dolora candida, is the horseshoe crab's obligate parasite. It spends its entire life on the horseshoe crab's appendages and book gills. As you can see in this one photo, there's congregations of them on the joints. And it lays cocoons within the gills and just spends its life within the gills and on the body. So to assess these, I asked a few questions. I first wanted to just get an idea of the population in general on the island. Also, I wanted to see if there was a decreasing gradient northward into the bay, because it is believed that during the off season, when they're not breeding, horseshoe crabs migrate into deeper, cooler waters. So my hypothesis was that if they're migrating inward, they would congregate more on the southernmost beaches instead of traveling up northward. So I looked at 3 sites here, T-Wharf being the name of the southernmost site, Chaseway being the middle site, and the north end, which is the northernmost site. I also wanted to see if the males and females were equally distributed. I figured it was important to determine that if you're trying to get an assessment on breeding individuals, you kind of want equal numbers of both males and females present. And to determine the parasite presence, I wanted to get an idea of the prevalence in Rhode Island of the parasite infestation. And I wanted to further determine if it differed between males and females, or if it differed across those 3 locations. So to answer these questions, I monitored the horseshoe crab population during those three locations during the new and full moon cycles at high tide. So I would go out 2 days before the moon cycle, during the moon cycle, the target moon phase, and 2 days after the target moon phase. And I would also sample the freely swimming individuals for the presence of the parasite. I would inspect their underside, flip it over like I do in this photo, and check for presence of the parasite by moving around. Well, they usually move around their limbs themselves, but you could just see if they're there because they're pretty big and visible. And I would also measure the curved carapace width at the widest part. I did not break up spawning individuals that were actively breeding because I didn't want to hinder the egg laying process. So I only sampled individuals that were just loosely swimming. Some of the findings that I found, there were a total of 106 horseshoe crabs over the 4 moon cycles. There were some challenges accessing all of the sites overall. So I broke down the crab counts per night per site. The most crabs were actually seen in the north end, the least being at Chaseway, which is by the Tidal Creek in the middle of the island, and the remaining was at T-Wharf, which is that southernmost site. So from the data, it wasn't suggested that there was that gradient as I originally hypothesized. Males and females seemingly were equally distributed overall across all the sites. There was 55 males to 51 females. However, I did notice that majority of the males were found in the north end and majority of the females were found at the southernmost site at the T-Wharf. The parasite findings, I found out that there was an 86.1% prevalence in the adults that I sampled. The data does suggest that it does differ between males and females based on their size, which is their curved carapace length, and that it does differ by location. For analyses purposes, Chaseway was not included in the location because there was only one of each sex monitored. So I only included Dwarf and the North End in those analyses. So in conclusion, overall, there was no northward gradient observed. However, I did reach out to the Rhode Island DEM's data set, and the gradient is seen there between these 2 sites that they monitor during the same times as I did. The northernmost site in the bay had, for this year, 36 individuals per night, and the southernmost site here, which just is on the cusp of Rhode Island and Connecticut border on the Atlantic Ocean side, was 120 per night. And when I looked at their 20-year trend, the southernmost site had on average 158 per night, and the northernmost site in the bay had 20 per night. So there's a little bit of a gradient seen there when you're comparing actual Atlantic to Northern Bay. And I also reached out to Justin and Chris at SoMAS, who were also doing studies on horseshoe crab populations there and parasites. And it differs from their study that they determined there was 100% prevalence of the infestation in the adults. And they also had the opportunity to test for juveniles, which they had 0% prevalence. I was not lucky enough to come across any juveniles while I would be searching, but in the future, that'd be great to study if I do come across juveniles. In conclusion, the New England populations seem to follow still a discrete estuarine populations. They don't believe that the individuals do migrate into deeper offshore waters within New England, and that Long Island is the northernmost limit where they do migrate offshore. So perhaps the small numbers of individuals within the bay are reflecting of that, and the difference in prevalence between Long Island studies and the ones in Prudence Island might be reflecting of less population and just less interactions with parasites among individuals. And I just really quickly wanted to acknowledge Elizabeth, my roommate and coworker, who helped me and was out there every night with me monitoring these crabs and the horseshoe crabs. Justin and Chris for all their guidance and advice in multiple Zoom meetings. And Charlene, my wife, for assisting when she would come visit on the weekends. And our dog, Christoph, who had no idea what's going on, but also enjoyed being there for moral support. Any questions?
Thank you, Kayla. Questions?
I have a question, of course, about the horseshoe crabs. What do you think like your total sample size was?
Just the breeding individuals, or just
Yeah.
the ones well, there's 106 total.
Okay.
I don't like just counts of just like actually actively breeding. I didn't sample the ones that were visibly nesting by like being dug into the sand or attached. But I don't have the number off the top of my head on the individual sample number that I actually checked for the parasites.
Kayla, I have a question actually about the coyote and the wildlife cameras. You
Yeah.
discussed. Coyotes on an island like that are a big deal in terms of what effect on other wildlife. Do you know, or does one know when they made it there, how they got on the island, what the population is?
Yeah, so we don't know what the population is. That's why the researchers set out to wanted to set up this study. It was primarily run by foxes before distemper wiped them all out. The deer population is thriving, but the coyotes this year, and we did start it because this year there were a lot of concerns from the residents because they would hear a lot of commotion in the woods. So we were like, let's try to determine that. But the island was primarily used in the 1800s, and a lot of people lived on there back then. So it is believed that they could either swam, which is what one of the researchers guessed, along with the deer. The deer, they guess, just swam over from the mainland, which would be kind of funny to see, or that someone had brought it over for means of anything with their farming, but most likely they believe it was from just swimming there.
Or during ice over winters, they could probably.
Yeah, that's true. Yeah.
Are there other questions? If not, thank you, Kayla. And we will go to our next speaker. Kayla, unshare. So the next speaker is Feng. The title of her talk is Developing Online Research and Education Programs During a Global Pandemic.
So my name is Jane. I did my internship with Cornell Cooperative Extension. So obviously since the coronavirus happened, I wasn't able to, go in personally. And they had to shift their focus to more online-based programs. So that's where I did most of my work. I did my internship with Hazel. She's a fellow CCE marine educator as well as an MCP alumnus. So the goals of Cornell Cooperative Extension is to pursue economic vitality, ecological sustainability, and social well-being. And they do this by bringing local experience and research-based solutions together, helping New York State families and communities thrive. My personal goals were, after completing this internship, was just to gain more personal experience with public education and get experience in creating my own project. I was also hoping to become better at communicating science to a diverse audience, because in my eyes, the engagement of the public is crucial in solving environmental issues. Obviously, this is true for all sciences, but I feel like it's especially true for all aspects of environmental science. Also, educating the public on local environmental issues will allow them to make the best decision possible when it comes to policy decisions, even though my internship is not really focused on policy. So in order to adapt to the situation and the shift from in-person efforts to online efforts, CCE created the Digital Education Initiative. This program is aimed towards K-12 audiences and meant to educate the younger generation. For this, they focus on a new topic each week, and for each week they post like infographics, activities, worksheets that the kids can do to learn about these topics. And so far, they've done they focused on water quality, seahorses, clams, alewife, and osprey. So this summer I mostly assisted Cornell Cooperative Extension with these ongoing projects and helped them compile information and format species identification cards and infographics that could be sold or put on their website or in boxes that they sell. I also submitted potential topic ideas for their future digital education weeks, and after discussing with Hazel, she thought that the best topic to focus on next was the invasive species, since it was more aligned with the work that CCE does. CCE also wanted to make a second kit after their first shell kit that they released a few months ago. And for the second kit, we decided to focus on Long Island's local seahorse, which is the northern line seahorse, and the eelgrass habitat. So for these, I also assisted in making thinking of activities, worksheets, and creating infographics for these things. So for the invasive species week, our goal was to make the issue more widely known, as well as highlight the impacts that invasive species can have on the local environment, especially for Long Island, which does depend on the local species for its economy. We also chose to focus on just the local invasive species as opposed to all invasive species. And the main reason behind this is that I feel like people don't really realize that these environmental issues can affect their personal lives. And because even with as big an issue as climate change, people understand that it is a big issue, but I feel like they don't understand that it affects them personally. And for the seahorse kit, we decided to focus on the local northern line seahorse, and the general goal was to raise awareness of the importance of the eelgrass habitat and use the activities to build a stronger connection between the people who purchased this kit and their local environment. And to do this, we included several outdoor activities and interactive science and art activities that were made with the help of Carolyn Monaco. She is a fellow marine educator and artist that works with CCE. They were also included because in previous studies, parents' response to these activities were overwhelmingly positive. Several had stated that this was like the first time that they had ever had the chance to do any hands-on experiments themselves. So for these activities, it's also a great way for the child to get the parent involved and interested in the environment as well. So this is just an example of one of the pages that was included in the booklet that was put in the seahorse kit. So it includes scientific information on the special traits that the seahorses have and the reasoning why they have that trait. And at the end of each little section, we've included a question and activity that kind of forces the audience to apply what they've learned from what they just read, as well as do an activity that can help them connect to the species more. So for camouflage and mimicry, we explained what it is, why they have it, and we asked them to think about if they can think about any other animal that also has this trait, and then to try and have them camouflage in with their own surroundings. This is just some more examples of the last slide continued. So some other traits that we included were the seahorse's independently moving eyes, suction feeding, and prehensile tail. So this is one of more examples of the last, more of the science-based activities that were included in the booklet. While we were originally hoping to include an activity where they could make their own seahorse seine and go seining and record data on a data sheet that we would provide with them themselves, with the help of and record species that other species that they found in the eelgrass habitat, we figured it would be unwise to have people go out around the eelgrass beds and disturbing the habitat without, the assistance and supervision of a marine scientist. So this is the alternative that we thought of. This activity allowed them to learn how to sex the individual, as well as take scientific data on likely the age and how big it was. And this is all obviously scientific data that scientists would take in real life. So this is the second page to that. So as you can see, all the pictures included are up to scale and a ruler is included. So for each one, they can measure the length, determine if it's of male or female. And this is another activity that was included in the seahorse kit. We were thinking of ways that they could help restore the eelgrass habitat. And so this is what we thought of, that they may help in the restoration of the eelgrass habitats. This is one of the easiest ones for them to execute, as well as had the least amount of risk as well. So what they can do is they can take the burlap that was included in the seahorse kit and follow the instructions. And once they finish making them, they send it back to the address that's provided. And then these are then used to plant eelgrass. So in this kit is also a survey with a series of questions that they can answer online. Unfortunately, since the seahorse kit is still under production and under the pre-order period, I actually don't have any data that I can analyze, but I do fully intend on looking at this data in the future when we do get it in. CCE can also use this survey to help cater and create their next kit. And these are just some questions that we've included, like favorite activity, how likely they are to purchase this kit in the future, and to recommend buying this to a friend. So this internship will help further marine conservation in that it is meant to educate and inspire the younger generation about this topic and inspire them to hopefully become marine scientists in the future. Their excitement may also transfer to their guardians and make them more aware of local environmental issues. And the National Science Teachers Association stated that, confirmed that learning science and engineering practices at an early age can foster a child's curiosity and enjoyment in exploring the world and lay the foundation for progression of science learning throughout their entire lives. There was also a study that found that a majority of scientists believe that the main barrier to a better understanding of science among the public was a lack of education, which shows that scientists should be educating the public more and engaging with them. Just some complications. While I was certainly hoping to find something that would give me more in-person experience, obviously the circumstances this year posed a challenge in finding an internship that allowed this. And also as a result, CC has had to cancel their usual marine science camp, which I'm sure what I would have been doing if, we could. So obviously there's a shift towards online outreach. Also, it did also limit the amount of hours that I was able to do. But yeah. So in conclusion, the work that I did with Cornell Cooperative Extension was with their digital education program. And from this internship, I learned how to more effectively communicate to the public, because even though the target audience of these was children from K1 through K12, it's still done in a way that allows any audience older than that to still learn some new things and work together with the younger generation. I also did communicate with other members of CCE about these projects, and so these skills will be easily applicable to any future career and not just any education-based marine science.
Thank you, Jane. We have time for a few questions. Not everybody at once now.
I have kind of a tough question. Well, maybe not a tough question. Jane, you did a very nice job on your presentation and also on the work that you did, especially considering that you had to do this online and with a program that traditionally hasn't had to do that. The one comment that I wanted to make, not a question, is about the tortilla. So for the Shinnecock Bay Restoration Program, we tried that technique. I think that we were following what Cornell had been doing. And we found out that most of those tortillas with the eelgrass, they didn't survive and contribute to restoration. And what we have found is that, so, we did that for a couple of years, and with such poor results, we decided to try something else. And what we have been doing is spreading eelgrass seed. And just like some plants do better if you plant them from seed, and they don't do so well if you try to transplant them, the seed was much more effective. And the third thing we found out was that, really the water quality, if it's good enough, the eelgrass that's in the environment will, on its own, reproduce more. And so anyway, I'm sorry to see that Cornell is still using the tortilla method, which has not been effective. Not your fault.
You said you guys used planted seeds?
Right. So these were taken from eelgrass, reproductive shoots, in the same environment.
We did consider that, but we figured it was the easiest to just do this because not everyone can go out and do it. So this would allow them to do it at home and just mail it in. So the seed was considered.
Yeah, it is harder to do a public event with seeds. It's definitely harder. But it's more effective.
Any other questions? Okay, if not, thank you, Jane.
Thank you.
The next speaker is Robert Rodriguez, speaking on piping plover conservation on Fire Island. And I see Robert is here.
Yeah. All right, can everyone see me?
Yep.
Yep.
All right. So yes, I'm Robert Rodriguez, and I did my internship this summer on piping plover conservation on Fire Island. So just a couple sections I'm going to take you guys through today. First, I'm going to provide some background on piping plovers. Then I'll go through the specifics of my internship, followed by the results, and then finally the conclusion. All right, so first, some background information. So nesting areas and migration patterns. So the piping plover is a migratory shorebird, and every summer they migrate north to coastal areas to breed. So there are three main populations we have here in North America. It's the Northern Great Lakes population, the Great Lakes population, and the Atlantic Coast population. Piping plovers tend to nest in sandy, shelly areas around the dunes. And as we can see from this picture on the top right, their eggs and their chicks blend in very nicely, so it often makes the nest hard to see. Piping plovers usually forage on the shorelines for marine worms, insect larvae, crustaceans, and other small arthropods. During the 20th century, the piping plover population suffered a sharp decline after being overhunted for its feathers, which was used in decorative women's hats, which we can see here in this image on the right. The Migratory Birds Treaty Act of 1918 provided some relief for the species until the 1940s. During that time, following the decades after World War II, a lot of traditional breeding grounds were lost due to an increase of commercial and residential developments. Coastal developments also attracted people to the beaches and other coastal areas, which caused a lot of the nests to be lost due to human disturbances. The species was eventually listed as endangered in 1986. Today, human disturbances still play a large part in hindering the recovery of the population. When areas around a piping plover nest is constantly disturbed, the adults get stressed out, and then which can lead them to abandon the nest, resulting in a failure of the nest. Although most humans unknowingly enter these areas with usually innocent intentions, the piping plovers still view them as predators and still can cause them to be stressed out and abandon the nests. As you can see from my little cartoon here in the top right, the species that threatens the piping plover most is humans, because in addition to coming up and physically disturbing nests, humans also have a tendency of letting their dogs run loose on the beach, which is also very bad for piping plovers. Flying drones and kites can also be perceived by piping plovers as predators. And then, of course, we have the natural predators of piping plovers, which are beach foxes, cats, and raccoons. And here we have some conservation methods that I used this summer to help the species. So the first is symbolic fencing. So what symbolic fencing does is just a very simple string fencing, but it's used to identify piping plover areas and deter the public from entering them, preventing disturbances from nests. Here in the top right, we have predatory exclosures, which are these big wire cages that can be placed over nests to prevent predators from predating the nests. As you can see here in the picture, there's little wire cells used to make up the exclosure, which allows the piping plover to move through the exclosure easily and keeping large predators out. Beach closures during the breeding season have also been a good conservation method, as a lot of the chicks were getting run over as they were moving from the shore, their nests by the dunes to the shorelines to forage. A lot of organizations do not take kindly to this beach closure, and so we have bumper stickers like this that came out that say "Piping plovers taste like chicken," which is very humorous. And of course, the best conservation method is always education, because by educating the public and spreading awareness of the piping plover and their endangered species status, it can prevent accidental disturbances from occurring at all. So now some of my internship specifics. So for my internship over the summer, I served as a piping plover steward through the National Park Service on Fire Island. So here we have a map of Fire Island, and there are 7 main breeding grounds for piping plovers. So we have first the Fire Island Lighthouse, which I was responsible for, Sailor's Haven, Talisman Beach, Blue Point, which is not depicted on the map, Watch Hill, and then the Wilderness area, which has been split into 2, the East and the West, and I was responsible for the East. All right, so the breeding season can be broken up into 3 time periods, and in each time period, the actions of both the piping plovers and myself tend to change. The season first starts off in early May with the nesting and egg-laying season. During this time, adults migrate back to their breeding grounds to reproduce. So during this time, the male really has 2 focuses. It's to establish their nesting areas while also trying to find a mate to eventually copulate and reproduce with. At this time, it is my responsibility to find the nests once the eggs have been laid. In the beginning of the season, I look for signs of that a nest or potential nesting areas by looking for courtship displays, which some of them are depicted in the left graph we have here. So one courtship display is the female will actually go under the male and pick at the feathers under its tail, which is depicted in image C here. And another courtship display is the high stepping tattoo dance, which can be seen in image D on the left. So another telltale sign of potential nesting areas are scrapes, which can be seen in the top right image here. And so these are caused by male piping plovers digging their breasts into the ground while using their legs to kick sand from behind them, which makes a little shallow depression, which will eventually serve as the nest for their eggs. Yeah. What's really cool about these guys is that once the last egg has been laid or the clutch has been completed, if you add 27 days to that clutch complete date, you'll have the estimated hatch date. And these guys almost work like clockwork. So 27 days after that clutch complete date is almost when they'll hatch. Of course, it's not perfect, but there's about a standard deviation about 2 or 3 days. And then from that estimated hatch date, if you add another 25 days, you have the estimated fledge date. So that's really helpful for me later in the season when I'm trying to keep track of which nests are hatching, which nests are fledging. All right. The next time period is the incubation time period, which ranges throughout June to early July. During this time, adult piping plovers will take turns incubating their nests, keeping their eggs warm while also protecting them from the elements. During this time, it is my job to make sure that the nesting plovers aren't disturbed and that the nests are not abandoned or predated. If predatory tracks are seen around a nesting area, then it is my job to go and erect one of those exclosures that I had previously talked about to prevent them from being predated. To deter larger predators and intruders, piping plovers have no real physical capability of doing this, so they tend to lure predators away through distraction displays. And so one of the most common ones is the broken wing technique, which I'll show right now. My camera. I didn't think you'd be doing this. All right, and as you can see from this video, the plover acts like it's hurt to draw in the attention of, there we go, to draw in the attention of predators, to draw them away from the nest using themselves as bait. Not the smartest method, but I guess it's somewhat effective. All right, so the final period is the hatching or fledging period, which runs throughout July to early August. So during this time, adults are protecting their broods or their hatched chicks until they're old enough to fly and become fledglings. During this time, it's my job to go out to each nest to find the broods, so the chicks and the adults, and to keep track of them and keeping tabs on them to seeing which or how many chicks are lost throughout the summer. Another one of my jobs I do during this time period is to look at the bands on piping plovers. So here we have a picture of me in the field using a spotting scope to look at the 3-character tags on the adult plover's legs. So these tags are put on by different organizations such as Virginia Tech, who do a lot of work banding chicks, just to be able to keep tabs on them as they migrate back to the North America each summer. All right, next is my results. So for my job for the National Park Service, they actually put me in charge of creating some ArcGIS maps depicting the locations of each nest. Shout out to Bob Serrato, because I would have never been able to do it without taking your class. So here we have the data for one of my sites, Lighthouse Beach. So as you can see by this map, it's right by the Fire Island Lighthouse, all right? And we had 5 nests at this site. We had a total of 19 eggs, and out of those 19 eggs, 11 of them grew up to fledge and will eventually go on to become adults. So we had a 58% fledge rate at this site. And so here at the bottom in this little Excel chart, we have all the data that was also put into this map. So we have the fledge dates, if it was disclosed or not, the hatch dates, and we also have the tags, the male and female tags seen here. So my next area is the Wilderness East. And so for this, it's a much larger area, and we had 11 nests here. We had 44 eggs in total, and we had 29 of them fledge for a 66% fledge rate. So as you can see, in the Excel sheet on the bottom, we had one nest failed, nest 6A, which is due to predation, probably by a beach fox. And for nest 2A, we actually had some confusion, and two of my coworkers accidentally labeled the same nest as a different nest. So, whole big thing. All right, and here is the Fire Island National Seashore nesting pair and fledgling count ranging from 2005 to 2020. So each year, Fire Island goes out and they release kind of like a report on the amount of fledglings and breeding pairs. And so, as you can see, in 2015, they were not really doing good at all. There was only about 2 or 3 nesting pairs with only about 5 fledglings. And then as you can see after that, the population, the breeding pairs and the chicks begin to go up until it reaches 2020 season. So this season, I'm very proud to report that we had a record high number of nesting pairs and fledgling counts, with 29 nesting pairs and 66 chicks that fledged. So the conclusion. So the National Park Service determines or labels a season successful if there's an average of over 1.5 fledges per nest. So this season, we had 66 fledges and 29 nesting pairs, which gives us an average of 22.8 fledges per nest, which my boss from the National Parks told me was also a record high, leading to a successful breeding season. All right, so thank you for listening.
Thank you, Robert. We have time for a couple questions.
I have a question. It's Christine Santora.
Sure.
Hi, Robert. You did an excellent job.
Thank you.
I was involved with similar work in Cape Hatteras National Seashore back in the 90s, looking at sea turtle nesting success, and I helped out with shorebirds as well. And that leads me to my question, because I'm wondering, it looks like you have a nice time series at Fire Island.
Mm-hmm.
Do you know of anyone who is looking at plover data across seashores such as Cape Cod, Fire Island, places like New Jersey and the Mid-Atlantic down to the southeast, to just see what trends might be happening, geographically over time?
I unfortunately not, but that sounds like very interesting data. But no, most of my work is just focused on Fire Island and some other sites.
Oh, sure. No, I didn't mean to imply that you should be doing that. I was just hoping that there would be some communication between seashores, that maybe somebody was looking at this data collectively. But in any case, great job.
Thank you.
I can answer that question. Robert said the Virginia Tech is collecting all that data, and then also the Fish and Wildlife Services, U.S. Fish and Wildlife Service has all that data.
Oh, good to know.
Yeah, thank you.
Robert, I have a question. Do you or does the Park Service have, can they speculate on why this was a good season for piping plovers?
Actually, yes. So looking at the data, I asked my boss the same question, because as you can see, about 5 years ago, there was almost no pairs. And so her answer was really, we don't know, improved monitoring, I guess, and more conservation methods. And it's still an ongoing process to try to get more people out there, get more stewards out there. So hopefully it's been a result of the product of our work.
Okay, thank you, Robert. Nice job. The next speaker is Elizabeth Salzman, and I see her here, talking on stable isotope reveal dietary preferences of juvenile sandtiger sharks in New York. Elizabeth.
So my capstone project is titled Stable Isotopes Reveal Dietary Preferences of Juvenile Sandtiger Sharks in New York. A special thanks to Oliver Shipley, Jill Olin, and Mike Frisk for all of their guidance in the world of stable isotopes. So apex predators are essential to the balance and function of any ecosystem. They regulate prey species that occupy the lower trophic levels, Without apex predators to regulate forage fish populations in marine ecosystems, there can be a trophic cascade and subsequent collapse. Since the dawn of industrial fishing, apex predators like sharks have seen drastic declines. This, coupled with climate change, has put shark populations at great risk. An increased understanding of juvenile sharks is essential in aiding conservation efforts. For this study, we looked at juvenile Carcharias taurus, or sandtiger sharks in the Great South Bay. Areas of the Great South Bay are known to be seasonal homes to these animals. The Great South Bay provides shelter from predators as well as a variety of prey options, making it the perfect nursery for sharks. But there remains the question of the impact these sharks have on the ecosystem while they are there and what they feed on during their residence there. This is a great figure from Nolan, who projected trophic positions within the Great South Bay necting community. The sandtiger sharks are believed to reside at the top of the trophic system while they're there, with bluefish and other fish just below them. So our question was, what are juvenile sandtiger sharks feeding on while in the nurseries of the Great South Bay? For this, we use stable isotope analysis. It's a method of non-lethal analysis that analyzes ratios of specific isotopes, mainly 15N and 13C, in the tissues of the animals, because these isotopes are transferred in predictable amounts from prey to predator. Delta 15N provides insight into foraging ecology and can help infer trophic position and food web structure, while Delta 13C provides insight into primary production and can help infer habitat use and movement patterns. Different tissue samples are used for different animals and for determining different things because of different tissue turnover rates, and these can be used in combination to indicate dietary shifts over time. This is the area of the Great South Bay where sharks were sampled from 2014 to 2019, and you can see in the bottom corner a little graph of the sharks from each year. It's proposed that they migrate seasonally through the mouth of the bay. These sharks tend to be found along the north side of Fire Island, particularly more in the sheltered areas like the little coves and marinas. Prey and shark data were collected separately. Prey data was collected in 2015 by Nolan. There were 19 different species that prey data was collected for, and they were split into 4 groups. For these animals, muscle tissue was analyzed, and then shark samples were taken from 2014 to 2019 by a joint effort between Stony Brook and WCS, and primarily blood plasma was analyzed for the sharks. Different tissues were used because of different metabolic rates in different tissues, resulting in different turnover rates. For the sharks, the blood plasma was used to give the most accurate picture of what these juvenile sandtiger sharks might be feeding on while they're in the Great South Bay. This graph is a biplot of the sharks' stable isotopes. You can see those values in purple. Those are the purple dots. And then the crosses are the 4 groups that I mentioned that we divided prey into, their values and their standard deviations. As I just mentioned, we split the prey into 4 different groups based on functional synonymity to allow for more accurate mixing models. Because mixing models only allow for a certain number of groups, we had to do this with the 19 species. The 4 groups we split them into were filter feeders, benthic invertebrates, forage fish, and larger fish. The main part of this project was analysis. So first I started off with gape width analysis, and then moved on to data analysis, and then we ran mixing models and estimated fractional diet composition based on those models. So for many animals, gape width limitation plays a large role in the size of prey that they are able to consume. Many sharks, like sandtiger sharks, are gape-limited species. Because this study looked at juvenile sharks specifically, it was important that we took into account the size of the prey that we were analyzing as possible diet items, as younger sharks will logically have smaller mouths than their adult counterparts. So as such, we constricted our prey resources based on prey size that would be in accordance with the estimated gape width. Typical gape limitations for most predators is about 10% of their total length, while some sharks have demonstrated larger gape widths. Studies also showed that gape widths proportionally increased with age, and as such, we kept our estimations for juveniles on the conservative side. So we kept it around 10%, but we rounded up to a prey size, maximum prey size of 15 centimeters. For analyzing the data and calculating averages and standard deviations, we started with that, adjusted for size, and then we ran the models. To analyze the data, we used a Bayesian mixing model because they allow for flexibilities in source numbers and for the incorporation of a number of uncertainties. This figure demonstrates a sensitivity analysis. The black dots on the figure represent the individual sharks and their stable isotope values, while the rings represent the different groups that we made. Because the majority of the shark individuals fell within the mixing polygon, it indicates that our identification of prey items was sufficient and we didn't leave out any major contributing diet prey items. The results of the mixing model gave us these graphs that demonstrate fractional proportion to diet for the 4 different designated prey groups. The peak represents 50%, or the median, which is the most likely contribution to diet. Larger fish showed the highest contribution, with forage fish having the second highest, then filter feeders and benthic crustaceans. These results support the idea that sandtigers reside in the top trophic position. The mixing models revealed that the median fractional contribution values for their diet to be 44.6% for larger fish, 37.2% for forage fish, 10.9% for filter feeders, and 7% for benthic crustaceans. This data provides valuable insight into the potential prey species that juvenile sandtigers are feeding on while they're in the Great South Bay. And these insights can help to better understand the role that these juveniles play during their seasonal residence in the Great South Bay. This information can be extremely valuable from a conservation standpoint. A major part of conservation of a species is regulation and understanding on what they are feeding on. Because if the fish that they are feeding on are decreasing, then the predators obviously will be decreasing as well. And an understanding of diet composition allows for better informed management and policy efforts. As long-lived species, recruitment and the conservation of juvenile sharks is essential to the survival of the species. As is common in the mysterious world of sharks, where not much is known, there's a lot of room for future research. Some suggestions for future research include investigation into the potential for unknown trophic competition within the Great South Bay that may play an unknown role in the trophic position and feeding for juvenile sandtiger sharks. Future research into the merit and method of lipid and urea extraction in sharks is also needed, as this method is currently used with other animals, but not suggested to be used in sharks due to uncertainties in the methodology and how well it works. Another area that would warrant future research is longer-term analysis of the diet of these juvenile sandtigers and their trophic positions and potential diets when they are outside of the Great South Bay. Stable isotope analysis has many challenges and drawbacks, but remains a valuable non-lethal method for understanding trophic distribution and diet. This study allowed for increased insight into the potential diet composition of these apex predators during their seasonal residencies in the Great South Bay, and opened a window to better understanding their ecological role while in the Great South Bay. This kind of information can hopefully help better inform policy and management in the future. Okay. Questions?
Questions? Elizabeth, what did you mean by the lipid and urea analysis? I wasn't quite sure what you were referring to there.
So in other animals, they will extract lipid and urea from the samples because they can have effects on the values that aren't necessarily accurate. But in sharks, they don't recommend it because the methodology has just not been researched enough.
Okay, thank you.
Good job, Elizabeth. I'm wondering if you thought about publishing this, trying to publish this work in a journal.
Oliver and Jill were talking about publishing it at some point, and yeah, they're looking into that.
Elizabeth, what is the population level of sandtiger sharks in Great South Bay now?
So right now, the population level is a lot lower. We were actually discussing this yesterday. So maybe 40 years ago, 50 years ago, the juvenile sandtiger sharks were basically overrunning the Great South Bay. They were almost like a nuisance species, and they really were the apex predator. And now their numbers are so much lower that if you look at biomass and all of that, they almost don't really reside in the top trophic position as they once did because their numbers have declined so much.
Okay.
Okay, one more question from me. In one of your earlier plots, it looked as though you had different colors for different years. Oh, sorry. It looked like the diet of the sandtiger shark might have been different by year.
For, let's see. This one?
It was before that. Oh, no, yeah, right. In the lower right-hand corner. So like all the grains, which is 2016.
Yeah, I mean, anything's possible. It could have to do with, you know, the brown tides or anything that were going on during that year in the bay.
Mm-hmm.
But for the most part, I think it was pretty concentrated in the center here. But 2019 and 2016 do seem to have some outliers.
Yeah, that might be worth following up on.
Yeah, that might be something to look into.
What's different about those years? Maybe the prey assemblage is different or something like that.
Maybe.
If there are no further questions, thank you, Elizabeth.
Thank you.
So the next speaker is Christine Suter. I see she's here, talking about sea turtles, debris ingestion and entanglement in coastal waters of New York.
So I'm Christine Suter, and today I'm going to talk to you about sea turtles, debris ingestion, and entanglement in the coastal waters of New York. This was an internship project that I did with the Atlantic Marine Conservation Society, which is based out of Hampton Bays, New York. They're also known as AMCs. Their mission statement is promoting marine conservation through action. And they do this in a number of ways. Just a couple of ways in which they do this are by responding to and performing necropsies on stranded marine mammals and sea turtles, conducting population surveys like surveys on seals, and hosting a variety of community outreach events like educational lectures and beach cleanups. Here's a photo of me responding to a deceased loggerhead turtle on Gilgo Beach this summer, taking what's referred to as level A measurements. So the big part of what I did for AMCs was the sea turtle data analysis project. For this, I was analyzing sea turtle data on marine ingestion and entanglement in fishing gear for the years 2010, 2012, and 2016. AMCs is part of NOAA's Sea Turtle Stranding and Salvage Network, and they have stranding files from as far back as 1980. And really, the last published study on sea turtles and marine debris and entanglement in the New York area was from back in the late 80s, 1989. So just to give you an idea of the sea turtles that we see in New York, there are 7 species of sea turtles worldwide. 4 of them we see in New York. First, we have the leatherback, which is the large one up on the right, Dermocoles coriacea, which is listed, actually all of these are listed on the IUCN, the International Union for the Conservation of Nature's list of threatened species. Leatherback is listed as vulnerable. We also have the green turtle in New York, Colonia mitis, which is listed as endangered. The loggerhead, Caretta caretta, also listed as vulnerable. And the Kemp's Ridley, Lepidopteris kempi, which is listed as critically endangered. So sea turtles are particularly prone to ingesting marine debris and becoming entangled in fishing gear because of their vast migratory routes and wide array of feeding strategies. All 7 species have been reported to have ingested marine debris or become entangled in fishing gear. And a recent global analysis estimated that about 52% of all sea turtles have ingested marine debris. Life history stage may play an important role in the likelihood of sea turtles coming into contact with and ingesting marine debris. Every turtle except the flatback turtle in Australia has both a neritic and an oceanic life stage. And the neritic life stage usually refers to like the coastal stage. And also diet and foraging patterns can shift depending on life stages. So juvenile and oceanic stage turtles are usually more likely to ingest marine debris than neritic stage. That's because they are usually feeding at the surface and exhibit less selectivity than neritic stage turtles. Oceanic stage turtles also pass through convergence zones, which are known areas of debris accumulation. So like, you know, the ocean gyres with all the plastic in them. So for the data project, the main idea was to review turtle data for 3 historical years. AMC's chose the years 2010, 2012, and 2016 because they provided a broad range of cases and represented major events with regard to sea turtles that had either ingested marine debris or became entangled in fishing gear. There was a total of 237 individual case files. That's a picture of me on the right going through just some of those case files. So I had to digitize all the data, put it all into an Excel database. And then the goal was to provide a baseline from which data in prior and subsequent years could be compared. And I was also trying to look for trends with regard to specifically marine debris ingestion and entanglement within and between those 3 years. So what I was looking for, some of the trends were percentage of each species that was involved with either debris ingestion or entanglement in each of the 3 years examined. I was looking for seasonal trends or time of year in which the events took place. I was also looking for the species in relation to type of debris or entanglement gear observed. And I also wanted to look to see if there were any location trends that could be found. So this map just shows all of the stranding events that took place during those 3 years, 2010, 2012, and 2016. And it shows the extent of the geographical range in which AMC's responds to stranded turtles, which is pretty much all of New York State. So the first thing that I wanted to do for the analysis was to determine what percentage of the strandings were somehow related to debris ingestion or entanglement in fishing gear within the 3 years and within each individual year. I also just want to make it clear that debris ingestion was confirmed by necropsy, and entanglement strandings involved any kind of fishing gear, whether it was active, passive, or discarded gear. So what I found was that 2010 really had the smallest amount of debris-related strandings, with only 1% and 9% for entanglement. 2012 had the highest amount of debris-related strandings at 15%. So then when looking at all 3 years combined, debris ingestion really only accounted for about 8% of the strandings and entanglement for 6%. So 8% seemed like a relatively small percentage of strandings involving debris ingestion. But I should mention that out of the 237 turtles, 43% of the turtles were not necropsied. So there's a very high likelihood that the percentage of debris ingestion is higher than 8%. So with seasonal patterns, the peak month for debris ingestion was during July, and that actually involved all loggerheads. And that sort of makes sense given that the loggerheads are most abundant during the summer when they're found foraging in the waters of New York. When I was looking at trends in debris type, the most common type of debris found was clear, soft plastic. The second most common, and that was found in 3 out of the 4 species. The second most common type of debris, which was found only in the loggerheads, was latex balloons. And they were actually balloons of all different colors. So here you can see up on the right is a photo of stomach contents from a loggerhead, which included some of that soft plastic and blue balloon. And then on the bottom was another loggerhead that was found on the beach, actually with a plastic bag coming out of its mouth. So there are a lot of different theories about why turtles ingest debris such as plastic bags. The biggest one is that it's been suggested that turtles eat plastic bags because they resemble jellyfish, and a lot of turtles eat jellyfish. But that doesn't really account for why they also eat other kinds of plastic like latex balloons and fishing line. So actually a very recent study came out that demonstrated the turtles may actually be drawn to the smell of biofoul plastic. So apparently organisms that contribute to the biofouling emit the same odor that comes from these oceanic foraging sites with high productivity. So it was unclear for most of the strandings whether debris ingestion was related to the cause of death, with the exception of one case, which was a male loggerhead that had ingested one of these Placker's dental flossing tools, which actually perforated the intestine and caused death for the animal. So I plotted the stranding locations in GIS to look for trends in debris ingestion. And what I found was that the majority of the debris-related strandings happened off the south shore of Long Island, in particular, all of the loggerhead strandings. It's hard to say where the loggerheads were, though, when they actually encountered or ingested the marine debris, because they can have a gut passage time of between like 5 and 23 days. So moving on to entanglement trends. As for seasonal patterns in entanglement, the peak month for strandings involving entanglement occurred during August. Leatherbacks were involved in the highest number of strandings, loggerheads with the second highest number. So as for the type of gear type, that was involved, passive or anchored gear were responsible for a majority of those leatherback strandings. Specifically like pot fishery lines and gill nets, and all three of the loggerhead entanglements were caused by hook and line. So this is just to give you a visual idea of what some of the entanglement strandings look like. The image up in the right corner represents the most common entanglement pattern for leatherbacks getting caught in pot fishery lines. So basically they bump into the line and then they start thrashing around so that the line becomes wrapped around both flippers and the neck. And up on the right was one of the strandings I reviewed from 2012 that exhibited that identical pattern of entanglement. So then we also have a loggerhead here, which just shows one of the hook and line entanglements. It had a hook, hooks caused in, hooked into two of its flippers and wrapped around another one of its flippers. So luckily, out of the 3 years of data I was analyzing, entanglement only caused the death of one animal, and that was one of the loggerheads in hook and line. So again, I plotted the entanglement stranding sites in GIS, and what I found was that most of the leatherback strandings happened toward the eastern end of Long Island. I think definitely adding more than 3 years of data to the map would provide a clearer picture of a pattern of stranding sites. And, unlike the debris ingestion strandings, for which we don't really know for sure when the animal ingested debris, the entanglement strandings provide a little more of an accurate location of where the turtles interacted with fishing gear. So pinpointing those sites where the turtles are interacting most with that gear might provide a tool to help mitigate future entanglement-related strandings. So just some closing thoughts about everything. Debris ingestion and entanglement were pretty much involved in a relatively small percentage of strandings overall. This could be good news, but it could also mean that we're not getting the whole picture, especially with regard to debris ingestion and, given the amount of turtles that were not necropsied. Also, definitely incorporating several more years of data would paint a clearer picture of these challenges that the turtles are facing in New York, especially with regard to these spatial trends in debris ingestion and entanglement. And I felt that one problem we may be able to tackle locally is the problem of leatherback entanglement and pot fishery lines. Perhaps it's time to start a conversation about the alteration of the gear so that it's more visible, or deployment of the gear at a time when foraging is not as high. This was done actually successfully with crab pot fisheries on the eastern Scotian shelf of Canada. So that is all I have for you today. And I did just want to say thank you to everybody who helped me along the way with this. So all of my supervisors at AMC's, Jennifer Lopez, Robert D. Giovanni, and Kimberly Durham, to Professor Ellen Pickich, my advisor for this project. And also thank you to Professor Bob Serrato, who helped me in my darkest hours of trying to get GIS to read my data the way that I wanted it to. So that is all that I have. Thank you.
Thank you, Christine. We have time for questions. Christine, have you looked at data from other regions other than New York to see whether the interannual trends that you saw in New York correspond to similar trends elsewhere?
I didn't, but I think that would be also a good next step for the project if I were going to continue to work on it. So, but yeah, it was mostly too because this was the data that was available at the time.
Yeah, Christine, you did a great job. Really interesting project. Just a comment. I was shocked at how important plastic balloons were in causing mortality of sea turtles. So
yeah, so
balloons at SoMAS events.
Yeah, I think it's a big thing lately too. There's a lot of drive-by birthday parties and things, and I actually, have been seeing a lot of balloons flying over my house lately. So not a good sign. I know, New York is kind of working on banning them altogether. I think it is still legal in some parts of the state to release up to 25 balloons, which doesn't make any sense to me.
Wow. Yeah, I really think we could do something about.
Yeah.
You mentioned latex balloons, but I thought Mylar
were the best. That was surprising too, actually, because it seemed to be all latex. So, yeah.
Glenn, this is Mike Frisk. Can I make a comment? Or?
Absolutely.
Yeah, I don't have a question, but because of this topic and Ellen's question, one of the things that has shocked me spending time on the water in both the ocean and the Sound in New York is actually how many floating balloons you see. When we cross the Sound, we collect a lot of balloons. I mean, it's absolutely amazing how many balloons are floating in the waters around New York. So this is certainly an issue. And anyhow, I've just been surprised by how many I've seen.
Thank you.
And then I have a quick question for Christine. I was wondering if you saw any research or when you were talking about geysers or pinch points for plastic collecting, is that occurring off Long Island and off the East Coast, or is that?
I mean, there is, the biggest areas of accumulation are in the ocean. Of course, there's always coastal garbage runoff that comes in from drainage and sewage and rivers and everything. But it's kind of hard to pinpoint where they are when they're picking it up or when they're actually encountering it. But I didn't actually look at specifically the if you're asking the places where the garbage is flowing into the water.
Yeah, not specifically, but I was just wondering if anyone mentioned that there are pinch points or geysers.
Not specifically.
Hard question, but no worries. Thank you.
Okay, I think we'll move on. Thank you very much, Christine. The next speaker is Allegra Irvin. Title is an assessment of the collapse and field recovery of 6 marine species within the Northwest Atlantic.
All right, my name is Allegra, and over the summer, I developed an assessment of the collapse and failed recovery of 6 marine species within the Northwest Atlantic. Dr. Frisk and I developed this project to be able to utilize at-home literature during the lockdown times to develop an analysis of species that have undergone population collapse in hopes to aid in conservation and management of these and other potentially vulnerable populations in the future. Finfish have been historically believed to be more resilient to population disruptions and overfishing that other species like marine mammals and apex predators have been affected by. Particularly over the past 150 years, finfish have experienced exploitation from commercial fishing practices that have impacted their stocks heavily and has led to federally implemented moratoriums, as well as led to programs such as seafoodwatch.org and other sustainability programs to work towards their conservation and better practices. These population collapses have endangered population management through shifting baseline syndrome and have established a new normal for what is considered a recovered or healthy stock biomass. The compounded effects of overfishing, key vulnerable life history traits, and shifting baseline syndrome can result in problematic or failed stock recovery. I compiled a review of 7 species that have been historically and presently exploited to the point of population collapse or functional extinction to highlight primary causation and ecological significance of these declines. I present here a series of case studies on Atlantic menhaden, Atlantic cod, Atlantic halibut, winter flounder, barndoor skate, and thorny skate to investigate their life history traits of these species as well as trends in their populations. For this presentation, I actually will not be reviewing my results for winter flounder or barndoor skate, mostly for time constraints, and the thesis is just as effective, or not just as effective, but effective without them for the time. So the first species that we're going to look at is the Atlantic menhaden. Because the species are short-lived, they're highly fecund, they're typically believed to be a little bit more resilient to disturbances. And menhaden populations appear to be abundant, but it's important to put these numbers in perspective and take their past population values into account when developing management and thinking about their populations. Valuable commercially as well as ecologically as a low-level prey species. Menhaden histories have been targeted for oil, for fertilizer, livestock and aquaculture feed, and most commonly nowadays, they're targeted for bait. An indication of the extent of their population decline goes back to the 50s and 60s, like I have here at 715,000 megatons. And then into the 60s, it went down to 162,000. So this is almost a 77% decrease in just 13 years of their biostock or their biomass. And these images I have here are from a paper published by the Atlantic States Marine Fishery Commission in 2020. So I have a couple more images, but I'm just showing them to you. So here shows an increase in fecundity and a decrease in fishing mortality. So based on these 2 images, it appears that the menhaden population is increasing or doing well. But then from the same paper, I have this demographic, which shows a consistent decrease in the young of the year abundance within the same time frame. Although the other age classes that you can see over here show quite sporadic abundance values, the green line depicting the young of the year shows a consistent decrease, which has implications for future populations of the stock. And then this is another graph from the same paper, a report. It shows increasing trends in menhaden landings between reduction catch, which is the industry for their oil and processing, as well as the bait catch, which is in the orange. So despite providing information showing increased fecundity and decreased fishing mortality, the same report also reported decreased young in the year abundance as well as increased landings. So it's kind of contradictory information. At the end of the report, they claimed that the menhaden is not overfished, although based on what they provided, I don't know how confident I am in that assessment. Menhaden fishery is a great example of a population that could benefit from a better catalog of historic population abundance and a better understanding of their ecological role and target population size. Despite being short-lived and highly fecund, the menhaden population still experiences overexploitation. And this is exemplified by the total allowable catch limit for the 2018-2020 season being set at 216,000 megatons, which is still a fraction of that 715,000 megaton 1956 landing size. So next I'm going to be discussing the Atlantic cod, which is another example of a species exhibiting traits that are believed to provide them protection from exploitation. They're early maturing, they're highly fecund, although I bet everyone's heard about the Atlantic cod fishery collapse, so these characteristics don't necessarily make them immune to overexploitation. So I'm going to use some data on the Atlantic cod to kind of show you an example of the shifting baseline syndrome, just because it's a good example of it. So you can see here, this is data from Morris and Green in 2014, and they were sampling in Gilbert Bay, Labrador, Canada. So you can see here from 2008 to 2011, their frequency distributions look a lot better. They're catching a lot more fish at a lot more variable size as opposed to in 2008. So if you're just looking at this, it appears that population is recovering or is doing much better than it was in 2008. But if you zoom out a little bit farther, back to their 1998 sampling, you can see that this increase is a little bit less impressive. So they were catching far more cod in 1998, and just the increase from the 2008 doesn't necessarily look like the population is doing great when you compare it to the 1998 model. So despite being a long-lived species that matures early and is highly fecund, Atlantic cod have experienced one of the greatest fishery collapses in the Northeast Atlantic. Regardless of any ecological influence hindering their recovery, Atlantic cod are in a permanent reduced abundance state that's going to make recovery of their historic ecological role very difficult or almost unfeasible. And this is just, you can see if they're looking a little bit more short term, the management is not going to be as effective, or it's not going to be as successful because they aren't looking to the right goal. So that's kind of how the shifting baseline syndrome affects management. So next I want to investigate Atlantic halibut. This species has unique characteristics, so it doesn't have all the characteristics that make it resilient or vulnerable. It's long-lived and late-maturing species, so these traits would typically lead a species to being overexploited. However, they're highly fecund, which influences their resilience by providing high reproductive output. Before halibut were targeted as a resource, they were considered a nuisance species, and here the success of the Atlantic cod fishery. So that just shows how abundant they were previously. However, by the mid-1800s, they were valued as more than bycatch, and the fishery declined significantly by the 1930s, and then it declined again in 1990. In 1994, catch management implementation, as well as a decrease in the use of otter trawls, is believed to have had a positive impact on halibut populations. So you can see by this graphic here, the decrease from 1985 to 1995. The stock stabilized by 2006 after the implementation of the management. And this graph is from Trzynski and Bowen in 2016. It exhibits the landings within the Scotian Self subject area. And the black line represents total allowable catch. So prior to 1994, there was no limit on it. And so you can see the decrease there when they implemented a total allowable catch. The same report claimed that Atlantic halibut on the Scotian Shelf and southern Grand Banks have recovered from their overexploitation. Again, here we see implications of shifting baseline syndrome as the populations in 2006 appear to be recovering, but they're still less than 50% their biomass in some of their history. I wanted to exhibit thorny skate because skate species exhibit different characteristics than flatfish and finfish, both in their ecology and in their life histories. They are late maturing, slow growing, and typically low fecundity. So they have all the characteristics of what you think of a vulnerable species. Thorny skate are managed under the Northeast Skate Complex Fishery Management Plan, along with 6 other skate species. These species were all historically classified as simply skate. So diving into the history of a lot of these species is a little bit more difficult because they were all classified together for a very long period of time. Despite protection under the skate complex moratorium, thorny skate have not recovered, although barn door skate under the same plant have shown increased populations. Thorny skate have seen significant and consistent reductions in abundance and habitat occupancy, with up to 95% reduction in abundance in large portions of its range since the 1960s. The continued decline of thorny skate exemplifies the faults in blanket multi-species complex management plans. And it's clear that the species requires new adaptive management strategies in order to recover. So through the research that I did on these species, as well as the barn door skate and the winter flounder, show that life history characteristics, something historically poorly understood in many finfish species, do not solely determine a species' resilience or susceptibility to exploitation. And without knowledge of historic biomass, nearsighted management, although well-intended, often leads to unsuccessful recovery. And this opened up a lot of thought for future studies, analyzing the life history traits and how they couple with exploitation to prevent population recovery. Also developing further case studies on other species, such as sturgeon, tomcod, Atlantic herring, and then using the information that we gather to investigate population dynamics of species that are currently undergoing a little bit of damage or in a little bit of danger. Evaluating specific species management plans, their successes, their failures, and using this information to develop better management strategies and try and avoid shifting baseline syndrome. So yeah.
Thank you, Allegra. Questions?
Allegra, this is Carl. Why did you choose the species that you chose rather than any other species?
So I was focusing on the Northeast Atlantic, and I was looking, it originated because I wanted to look at species that needed conservation or that were undergoing historic collapses. So that's kind of where we started with that.
Okay. So you were sort of geared to species that have been in trouble, basically.
Yeah, and using the information that I could glean from that to kind of work towards developing management strategies that would be a little bit more successful than in the past.
All right, thanks.
Any other questions or comments? All right, thank you very much, Allegra. Nice job.
Thanks.
The next and last speaker for the symposium is Jonathan Garr. I see him here, and I see Christine O'Connell, his mentor. Title, Interpreting During the Pandemic.
I'm Jonathan. This summer I worked as a park ranger interpreter for the Fire Island National Seashore. Kind of funny, growing up on Long Island, I was not even aware this was a national park or a seashore, even though it was established in 1964. So this was a very cool experience for me. To work here. It's one of 10 national seashores and one of 419 national park sites, which also include recreation areas, parks, and historical and cultural sites. The Fire Island National Seashore represents 32 miles of barrier island off the southern portion of Long Island. Next, okay, cool. There are 5 visitor centers that I was stationed at throughout this summer. So the first one is on the way west on the Fire Island Lighthouse. After that is the Sailor's Haven Visitor Center, which I spent most of my summer at. in the Watch Hill Visitor Center, the Fire Island Wilderness Visitor Center, and then also on mainland Long Island, we have our William Floyd Estate, which was home to a signer of the William Floyd, a signer of the Declaration of Independence. So there's a lot of cultural and historical significance of that site. So yeah, I when I accepted the position, you know, I expected to be given guided tours and walks through the forest, on the beach, in the bay. And I was also told I was going to do canoe tours, which sounded so cool to me. And we would also host large school groups throughout the fall, like as a field trip. But unfortunately, that all went on pause. So we had to figure out what we were going to do this summer as interpreters on Fire Island. So fortunately, our visitor centers remain open. Compared to a lot of parks in the area, they closed everything for the season. Yeah, we were open. I had a job. I was happy. But we had to find creative ways to interact and educate with the public. So basically, I would interact with the public from behind the desk, most of the time, I would say. You know, we would put up barriers, we wear masks, but we could still have really, you know, great conversations about Fire Island, about the beaches, about anything, you know, really. And then we also provided arts and crafts for kids, little activity kits. And we also have our Junior Ranger program, which is a big hit always. So every educational activity we wanted to do, we had to justify that we can our employees would be safe behind the desk or, you know, in the field. So and we would have to wipe down everything we touched. So that took up a lot of time. We kept our distance. We wore masks. But other than that, we definitely were busy with a lot of visitors, and it was good fun. So this is actually the Sailor's Haven Visitor Center. Like I said, I spent most of my time here. Unfortunately, we were not allowed to have visitors inside, but we would let them peek their heads in to look at the displays. We put up these signs everywhere. We really wanted our visitors to make each other feel comfortable and safe as they're on narrow boardwalks, on forest trails, in confined spaces. And, we would draw up signs like these. So another part of my job, besides interacting with visitors or walking or in the, interacting with visitors at the visitor centers, I would be walking around and interacting with them on the field. We call this roving. I would tell them not to touch poison ivy and watch out for ticks. And then we would also make short educational videos, which can be found on our Facebook pages and social medias. So that was a new experience for me. I never participated in video making. And other than that, the rest of my time I spent walking around all the beautiful places we have here on Fire Island. On the left is the Sunken Forest Trail, which is just amazing. I'll tell you about it later. Our beaches and our marsh. All right, and I also just, I love this stuff, and I found so many cool things and so many treasures. So that was really neat to me. So as an interpreter, my job was to educate the public. And the whole idea of interpretation is to connect the tangibles, which are, the physical objects in our world, so the animals, plants, and the weather. We wanted to connect those tangibles to the intangibles, which is the culture, relationships, and community. So for example, I could tell you all about how the piping plover is important and why we should protect it because of its important ecological role and that it's endangered. But I can also try to curate a story, or tell a story of how piping plovers are incredible parents and represent a close-knit family. So like Rob was talking about before, They do the broken wing display, which is kind of brave, I think. And they go to great lengths to protect each other. So the idea of telling the visitor this is that we want the visitor to make a connection to their own morals and their own life experiences to be able to relate to the piping plover family. We want the visitor to ultimately want to protect the piping plover through their own will or willpower, rather than just following the rules and regulations. So that was a new experience for me, and I learned a lot, and it was very fun. So yeah, during the pandemic, we wanted to, that's what we would normally try to do, and it was very easy to transition, I would say, interpretation skill set, I guess. But we wanted to educate the public, as many people as possible, but we didn't want to gather large groups. We wanted to provide the tools to learn and create, but we actually couldn't physically exchange or hand out items to an extent. And then we wanted to make the park visitors feel welcome, but there we were behind the desk in a mask, hiding behind a plastic shield or something. So it was kind of funny. But the visitors were, I got so many compliments just saying how happy they were that we were at least open. So that was cool. Yeah. How do you press this button? Oh, and yeah, this is our interior of one of our visitor centers. So normally the kids love it, I was told, and we couldn't let them in. It's devastating. So this summer, I was kind of interested in seeing what percentage of our visitors were under the age of 18. The park has never really recorded any data of this type, I guess. And so I was the only employee taking this tally, and it was, it was strictly visual, guessing below the age of 18. And the data was only recorded at the Watch Hill and Sailor's Haven Visitor Center because I spent most of my time flipping between those two. A total of 25 days were recorded, and only visitors who actually approached the visitor center were counted, not anyone who I was interacting with in the field. So it was pretty interesting to me and my supervisors. In total, the average was about 38 visitors at each visitor center. Sailor's Haven had more, and the percentage of youth under 18 approximately was about 29%. So that was very interesting to us, and we're in the process of comparing it to prior year's data. So yeah, as the summer went on, I wanted to get visitors more involved, especially at our Watch Hill location, as it does not have the sunken forest that Sailor's Haven does. The Watch Hill location is a very beautiful location. It's very buggy, so people just kind of go straight to the beach or the restaurant. So I really wanted to get them more, engaged in learning about the environment, even though the interpreter rangers were not really allowed to get close and educate them as we would normally have done. Like I said, the guided tours and canoe tours were canceled or on hold, on pause, and we had to keep everyone at a distance. So my idea was, or a couple of our ideas was to create an expedition for the visitors, which was really fun to create. It was basically a mix between a scavenger hunt and a self-guided tour, and the idea was to get the whole family, a whole family involved. And also this expedition would be available at all times, as it would be on the boardwalk permanently. You didn't need a ranger to complete the expedition, and hopefully you learn a few things along the way. So this is an example of our test run. We're in the process of getting more permanent structures, but it was a great test run. We got a lot of feedback. People thought it was great. So, this is kind of overwhelming, but basically each piece of paper had a fact about an object or an animal you could see in the marsh, in the forest, in the beach. And then that fact would relate to the next clue and help you or tell you how to find that next clue, and you follow the boardwalk and go to the next clue. So it was fun. The idea was for the child to follow and find the clues, and the parent to help them understand the concepts. And so yeah, as of now, I'm in the process of completing one at our sunken forest, another expedition. And then other than that, we're just manning visitor centers and creating more videos. At the sunken forest, I will focus on the globally rare maritime forest, which I will talk about now because this is what I talked about all summer, and I love talking about this forest. It's really a unique site. So I'll start with the first part. It's globally rare as it is only found here on Fire Island and in Sandy Hook, New Jersey. So this particular maritime holly forest is made up of 3 tree species: the sassafras tree, the juneberry tree, which is also known as the serviceberry or shadberry, and the American holly tree. So when I say it's globally rare, that type of forest community is not found anywhere else besides here and Sandy Hook, New Jersey. So it's really unique. It is thought to be approximately 280 years old, more or less, and it is only able to form due to the secondary dune system you see on the top center and left image, which is not that secondary dune system is especially prominent at Sailor's Haven. And it's not too common along the rest of Fire Island. And basically, that secondary dune system provides a secondary barrier for salt spray and high winds to protect the forest and allow it to grow. This forest is actually a very slow-growing forest. As you can imagine, there's a lot going against its survival, you could say. There's poor soil. It's just sand. It was just sand at one point. There's a thin layer of, nutrients from trees falling over time and leaves debris, but it's mostly just sand. There's a lot of salt water in the intruding inward, especially due to climate change and sea level rise, and then high winds and also salt spray. So that tree actually fell the other week. It's about 17 inches in diameter. And I don't know how familiar you are with trees, but I counted 150 rings, which annotates it's about 150 years old. And a tree 150 years old, it's a red maple, would probably be twice or even 2.5 times the size of that one that it should be. So that just indicates the rings were very slow or small, and the tree grows very slow. So that's very interesting to me. It's also highly desired wood due to the proximity of the rings for furniture making. So pretty cool. So yeah, it's not a friendly environment for a forest to thrive, but somehow it does it, and it amazes me every day I walk through it. And I think that is it. So yes, that was my summer internship work, and I had a lot of fun. And I want to thank you, everyone, for this semester, this year. Thanks, Christine, Dr. Christine O'Connell, for being my mentor, and all the SoMAS facility, faculty, and staff. Thank you.
We have time for a question or two.
Come out to Fire Island. I'll give you a tour.
Colin, and I'd love for you to just talk about some of the challenges you had with trying to revise how you do interpretation and conservation with the public, given COVID. I know you had mentioned even handing out pencils and paper wasn't allowed, things like that.
Yeah, so we weren't allowed to exchange items, but of course we can hand it out. But then it becomes a cost factor. But yeah, and then so I guess this is my first year, but last year they would have all these activity kits that we would give out to the public, and we would have the children come back and we would learn all about it in the visitor center. We would sit down, we'd have late night campfires. We could, in the guided tours, the guided walks, and it was just really hard to, my biggest thing is I wanted the visitors to still feel engaged and still be able to learn something from their experience here at Fire Island. And I think we did a really good job, but it was kind of a mad dash and a mad rush to get it all together for them, especially since we were delayed in our start. And then also, as I mentioned before, we had to justify every action and with a lot of paperwork, as it is the federal government, to prove that we would be safe ourselves, and we also, the visitors would be safe.
Anybody else? If not, that concludes the MCP Symposium of 2020. I'd like to thank all the presenters.
Woo! Yeah, great job, everybody. Before we close, I would like for everyone involved in the MCP program to unmute yourselves for a moment and to give Glenn Lopez a big round of applause. Director of the program.
Well done. Thank you, Glenn.
This is your last event as Temporal program director, and I think he served us very well. Thank you, Glenn.
Mm-hmm.